Of Chicago and completed his postdoc training in Eric Elm's lab at the MIT. His postdoctoral work focused on developing techniques to quantify individual-specific evolutionary dynamics within the human gut microbiome. He then came to us at the Institute for Systems Biology, and his lab develops computational and experimental tools for investigating host-associated microbial communities to explore interactions between ecology, evolution, and the ecosystem function. He is now working on developing personalized interventions for improving human health and well-being. So Sean, take it away.
Hello, everyone. A bummer that we lost Dr. Jimenez. She just dropped off our Zoom. I'll filibuster for a second here, just in case she pops back on as I pull my slides up. But rather than missing out on our second talk, I guess I'll give you all a little story here on the microbiome and its impact on how we respond to drugs. Let's see. No news. She's still not on. Okay. Okay. Well, sorry everybody if you're going to get me. Sadly, I had a backup talk ready to go. Are my slides looking the way they should look? They should be swapped. Okay. So this isn't perfectly in line with the theme of this session, although there will be some mention of Prevotella, and I guess we got a really great overview of the sort of curious case of Prevotella from Dr. Luzapon. So, okay. Recent work from our group in the past few months that was published in the journal Med has delved into this question of, we know the microbiome can metabolize drugs and can interact with pharmaceutical drugs, but how does that really impact the human side? How does that affect the response of us to those drugs? So we'll get into that. And I don't think we need much introduction on the microbiome, since we've gotten a fair amount of that. I like to say it's sort of changed the way we think about ourselves as entities, right? We're not entirely human. We're never alone, right? We're more than 99% microbe when it comes to the genetic composition of our bodies, right? More, but it's 100 times more genes pent up in our microbiome than there are in our own genomes. And it's a complex system, right? In complex systems, the whole is greater than the sum of its individual parts. And sort of classic 20th century reductionist forms of doing biology aren't as relevant to these types of ecological or system scale phenomena. There's a lot of emergent phenomena that can come out of these systems, and you have to study them at multiple scales and integrate across those scales in order to get meaningful insights out. So again, we know the microbiome is important. It affects all the systems of the body. You can kind of think about it as a metabolic or endocrine organ. It probably affects about half of the circulating metabolites in our bloodstream. But the question we're going to ask here today is whether or not it affects how we respond to drugs. And the drug in particular that we'll focus on are statins. Statins are cholesterol-lowering medications taken by lots of people. I think it's the most prescribed drug on Earth, definitely in the developed world. More than 20% of all Americans over 40 years old are taking these drugs. And they're life-saving, right? They reduce the occurrence of stroke and of heart attacks, and generally they've been a huge boon to medicine over the past several decades. Now, while they do save lives, and you should always listen to your cardiologist and take your statins if they tell you to take your statins, there is an interesting wrinkle to how statins affect our bodies. One of the most common side effects that I won't talk about as much is that they can cause muscle pain, pain in your muscles. And so a lot of people suffer from this, and they have to work with their clinicians to find a tolerable dose of the drug that allows them to take the drug without pain. But the other big side effect that we'll focus on more today is that it sort of messes with your metabolism a bit. It messes with how you process lipids, and this can affect your chances of developing type 2 diabetes. Those who are on statins are more likely over time to transition into type 2 diabetes. It sort of makes you more insulin resistant, essentially. And oftentimes people with diabetes themselves are targeted for statins, for statin use, because they themselves are more likely to experience heart attacks. So it's usually thought that heart attacks are scarier than diabetes. And so that trumps the fear of progressing towards type 2 diabetes. So usually people are just given statins without much thought to the sort of metabolic side effects of those statins. So this brings us to our study. We were interested in the heterogeneous effects of statins across a population. So this is work that was led by Tomasz Wolmanski and our collaborators lab, the Hood-Price lab, along with my co-corresponding authors, Noah Rappaport and Andrew Magis. Some of the microbiome data processing was done by Christian Diener, who you all know well at this point. And like I mentioned before, Nathan Price and Lee Hood are the collaborative lab that helped out on this project. So if you look at prior work that delves into the efficacy of statins, you see in this, I think it's called a waterfall plot, a huge range of effects to the same exact dose of a statin over the same time period. In about 8,000 people here, you see that some folks have a really, really strong response. Their LDL cholesterol is taken way down when they take a statin. But in some people, you actually see an increase in LDL cholesterol with statin use, which is not what you want to see. And indeed, the amount by which the drug brings down your LDL cholesterol, that is correlated with, or it predicts your risk for events downstream, for having a heart attack. So you can see that people who had the biggest reduction in LDL have the lowest risk, essentially, for cardiovascular events. There's been some prior work in the microbiome field in statins and the microbiome. This is a paper from 2020 in the journal Nature, where they were focusing more on the effects of the drug on the microbiome itself, right? To taking statins reshape the composition of the microbiome. And this is a European group that often works in what we call enterotypes, which is a sort of clustering method to cluster together similar types of microbiomes. And in the developed world, although this is definitely not the case in the developing world or other parts of the world, but in the developed world, we tend to see 4 clusters. There are 2 clusters dominated by the genus Bacteroides, which you saw in Dr. Lissabone's talk. This is the genus that is dominant when Prevotella is not dominant. There's a cluster that's Prevotella dominant, where Bacteroides is actually very low in abundance. And then there's a cluster where it's enriched more in Firmicutes, or in particular Rominococcaceae, so depleted in Bacteroides. So these 4 groups are distributed across the population. And what they found is that, this BAC2 group is associated with worse metabolic health. So people with higher BMI tend to be enriched for this particular enterotype. And the big take home of this particular paper was that statins appear to protect people from transitioning into this BAC2 enterotype. It was a cross-sectional study, so they didn't actually watch people transition, but they found that there was a slight depletion in this BAC2 enterotype in obese people who were taking statins. So there was an idea that there was maybe some protective effect here. We were actually more interested in the inverse question of whether or not microbes, the composition of your microbiota affects your response to statins, not how much statins affect your microbiome. In fact, we didn't really find much of an effect of statins on the microbiome itself in our study. The reason we embarked upon this question is prior work has shown that if you expose commensal microbes that are in the human gut to a wide range of drugs that humans take, different pharmaceuticals, you find that many of our commensal microbiota are able to metabolize known pharmaceuticals. And so this is just an example. On the x-axis are many different strains of commensal microbes that were cultured in vitro, along with a drug, in this case, lovastatin, which is a statin that I think is the original form of statin, which was derived from a fungus, I think. And you can see that by and large, organisms in the Bacteroidetes phylum tend to be pretty good at degrading lovastatin. And you see patchily other taxa doing it as well, but the take-home message is microbes can deplete the concentration of a drug if it's present in the gut lumen. And so what are the mechanisms that could be going on here? It's kind of hard to say. There's a lot of ways things could be acting, like the statins could be reformulating or reshaping the composition of the microbiome, which has collateral downstream effects. That's sort of what this other Nature paper was trying to point towards. Microbes can be depleting the amount of the drug that's available to the human body and pulling it out before it gets absorbed. And statins, their main effect is to inhibit an enzyme in the liver, HMG-CoA reductase, which is the rate-limiting step in cholesterol synthesis. And so maybe microbes can interfere with that inhibition by depleting the drug, or maybe even turn it up by turning the drug into a more active form, into like a prodrug. And another form of modifying the body's cholesterol, like Dr. Poirier mentioned, is that microbes can convert cholesterol into caprostanol, which maybe increases the removal of cholesterol from the body, or they can affect bile acid synthesis. And so that could also potentially be contributing to the balance of cholesterol in the body. So we are interested in kind of seeing the overall reflection of what's going on in the gut, with the ecology of the gut, the metabolism of the gut, how that's reflected in the body, and in this case, how it's mediating our response to drug. So, okay, here's the data set that we analyze, is this Aravel cohort, where we have a lot of paired information about host physiology, molecular phenotype of the human side, along with the gut microbiome. In this case, we sort of sub-selected a set of people where we knew their statin usage information. So we had about 1,600 non-users and about 250 statin users. And among those statin users, we had really high resolution information on the dosing of the statin they were taking for about half of those folks. And so again, statins are inhibiting this particular enzyme called HMG-CoA Reductase. It's actually hard to measure the effect of statins without having a time series. Usually the effect of a statin is measured as the change in LDL cholesterol over time since taking the statin. But we only had a snapshot. We only had cross-sectional data for most of our population. So we wanted a marker that was sort of a snapshot marker for statin efficacy. And we landed upon a particular chemical that we could measure in the blood called HMG, which is essentially this upstream metabolite in this pathway. It gets cleaved from the CoA by an esterase, and it just sits around in the bloodstream. And so we thought that, well, maybe people who had more or less inhibition, we'd see essentially the quantitative relationship with the levels of HMG in the blood. And yeah, we kind of did. It was a little noisy, but it seemed to kind of work. Where in people who were not taking statins at all, there was no relationship between LDL cholesterol and plasma HMG levels. But when people were taking statins, there was a negative association. So lower LDL cholesterol, more buildup of this precursor metabolite in the blood. And overall, there's a population shift towards higher HMG if you start taking statins. So now we have a measure of on-target efficacy of the drug. Just to delve a little more deeply into whether we think it's a good measure of this efficacy, we see that there's a quantitative relationship between statin dose and the level of this marker in the blood. So there's a positive correlation, right? The higher of a dose of statin you're taking, the more you see building up in the bloodstream of this HMG. And it's also associated with LDL cholesterol coming down, right? So higher dose is also associated with a steeper reduction in LDL cholesterol. There are also genetic markers of statin efficacy, SNPs essentially in your genome, that determine whether or not you're going to respond more or less to a statin treatment. And we see convergent evidence here as well for HMG as a marker, where we see no differences in plasma HMG levels between people with two different genotypes for high responder and not a low responder to statins when they're not taking the drug. But when they're taking the drug, you can see everyone goes upward in their HMG. Everyone has more buildup of this particular analyte. But people who are predicted to be stronger responders from their genetics see an even greater bump upwards. So this is just further evidence that HMG is indeed a decent marker of on-target efficacy. So what if we look at the microbiome? So when we look into the microbiome, we find that people who have more diverse gut microbiomes show less on-target effectiveness of a statin. So their HMG levels bump up not as high if they're really high diversity versus low diversity. So on the x-axis are the tertiles of Shannon diversity. So the most diverse third of the population, the middle, and the least diverse on the left-hand side. And for here we have, I think, both, richness, observed ASVs, and Shannon, and we see the same sort of effect. You can ask whether or not this is robust to dose, and indeed it is for the small subset of people where we have dosing information. And if we control for many other confounders, we find that there is a negative association between diversity and the buildup of this HMG. There could be a lot of reasons for this. We don't quite understand it yet. It makes some sense that maybe if you have more diversity, the chances that you have organisms that are good at degrading statins maybe increases. So let's go back to this enterotype picture. So in that prior paper, they had looked at these clusters called enterotypes. And we were able to cluster our population into the same four groupings. So that was convergent between these different data sets. Like we talked about in Dr. Luzapone's talk, there's a group of people who are highly enriched in Prevotella. And so that particular enterotype shows this really bimodal association. Either you have a lot or you have a little. In the Bacteroides groups, you see that they both are enriched for this particular genus. But this BAC2 group that's thought to be less than healthy, it shows a characteristic depletion in butyrate-producing Clostridia, like Faecalibacterium presnitzii, for example. So it shows the lowest abundance of this particular organism. Okay, so we did try to recapitulate this effect that was observed in the Nature paper, that maybe statins were somehow protective of the BAC2 phenotype. We actually could not find that result. We saw indeed the opposite, actually, in our population, where people who were statin users actually were enriched relatively to non-users for this BAC2 enterotype. And this was true even if we looked at the same data sets that this group looked at. However, if we partitioned our analysis and looked at people who were very sick, who had essentially advanced heart disease, those individuals did show a depletion in BAC2, but it only really worked if you subselected the population in that particular way. So this is where it gets more interesting, I think. If we then, instead of thinking about our statins affecting the composition of the microbiome, which in our analysis, they were, there was a significant effect, but it was very, very weak. What was much more striking was that the composition of the microbiome seemed to affect the on-target efficacy of the statin. So you can see this BAC2 group, they show the biggest jump in their HMG when taking statins compared to non-statin users. And the BAC1 group also shows the next biggest jump. The Ruminicocaceae group, they show a significant upward tick in their HMG levels when they take the drug. So they're having an effect. It's not as strong. And interestingly, we didn't have a lot of data for the Prevotella group. We don't have a lot of data points here, so it's hard to know whether this is a really reliable result. But it seems that those folks who have high Prevotella microbiomes didn't have as much of an effect when taking a statin. And this is also reflected in how much you bring down LDL cholesterol. The folks with high BAC2, they brought their cholesterol levels down the most. And this was all kind of controlled for a lot of different confounding variables. Now, what's even more interesting, I think, is that the off-target effects in terms of promotion of metabolic disruption, these were also affected by the microbiome composition. So people with a BAC2 enterotype, which tangentially, I should mention, Harkening back to the alpha diversity result, BAC2 is also the lowest diversity of these enterotypes. So the association with the diversity seems to be largely driven by this BAC2 enterotype. Those who have this particular enterotype show the biggest perturbation to their metabolism. So their HOMO-IR is elevated, their HbA1c is elevated compared to everyone else. So they seem to be most disrupted in their metabolism when taking the statin. You see a little bit of that in the BAC1 group, excuse me. But the Robrina-Caucasia group appears to be completely protected from this. We could recapitulate all of this analysis in an entirely independent cohort from Europe called the Metacartus cohort. Excuse me, I'm recovering from a bit of a cough. And we see the same exact results. So this bacteroides association is true in this other cohort. The enterotypes on-target and off-target effects were true as well, which is super exciting. So, this is, I think I'll wrap up with that. This is sort of the first, I think, detailed observation of the sort of host side efficacy of a drug being modulated by the composition of the microbiota. Oh, thank you so much. Here's my water. But we're trying to understand the mechanism still. We don't quite know what's going on. We have some data on blood metabolomics suggesting that certain bile acids in the blood might be modulating these effects. So we see, say, for example, ursodeoxycholate enriched in the bloodstreams of people who have high BAC2 enterotypes. But ultimately, we need to do more work to understand mechanistically what's going on here. And we'd like to run some sort of trial, either a longitudinal trial or even an interventional trial, to see if we can leverage what we've learned to give people precision drug interventions that are optimized for their metabolism and for their bodies. And that's ultimately the dream, right? To be able to leverage this information for precision medicine. So I'll skip this. Christian is actually currently working on ex vivo studies to try to get a mechanistic picture of what's going on. Thanks to the lab.